Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
And together with my colleague Mike Ziskowski, who will be joining me on stage for the second part of the talk, we'd like to tell you a bit about the Microsoft Biology Foundation. So the title for this talk is an open source library for bioinformatics. We didn't want to call it, as you'll see it's titled in the program, which is the Microsoft Biology Foundation, because we're going to change the name. So everything you hear about the Microsoft Biology Foundation today, from July, will be called Biodex. And there's reasons for that, which I'll explain as part of the talk. So anyway, what I'm going to tell you about in my half of the talk is I'm going to give you a little bit of information about the Microsoft Research Connections team. I won't duplicate too much the information you heard from my colleague Lee earlier. We should probably sync on our slides next time. But I'm going to tell you a little bit about the reason why we exist and the way in which we work. And then I'll move on to describe how the Microsoft Biology Foundation is an example of that way of working. And then I'll give you some information about MBF itself. Now, I don't plan to dive deeply into the code. On Monday and Tuesday this week, we actually held a workshop training some people, some of you are in this room today, on how to use MBF to do programming. But what we'll do is we'll cover it at a higher level. Now, all of the tutorial materials we delivered earlier this week are available online, and I'll give you some links to those if you're interested and weren't able to make the course. So, the Microsoft Research Connections team. We're spread around the world. We're located in all of the different Microsoft Research labs around the world. So there's a group in India, a group in Beijing, a group in Australia. There's even a small group here in Latin America, also Europe, and of course in Redmond in the United States. Not only are we distributed around the world, we're distributed around the different types of science that we believe Microsoft Research can assist in terms of the types of technology we can bring to bear to make that science move further and faster than it could have without the assistance of Microsoft. Those areas are natural user interfaces, earth, energy and the environment, health and well-being, computer science, and my area, and Lee's area, education and scholarly communication. Now, in each one of those areas, we work essentially in two different ways. One is that we establish collaborations between Microsoft and between academic researchers. Those collaborations all take the same essential form, which is Microsoft brings the computer science expertise, and the academic brings the expertise of their particular area of research. And together, we try and do something by applying the computing to that area, which could not be done before. Now, those collaborations are very hard to create because Microsoft researchers work with many different people, and it's sometimes hard to make those connections. But when we succeed in making one of those connections, sometimes the results are surprising, and I'll give you some examples in a moment. The other way that we work is we generate software. Now, you saw plenty of examples actually from Lee, and you'll see another example in terms of the Microsoft Biology Foundation from me today. So I'll just move on and I'll show you, the areas that we work in here. Now, this is within health and well-being, and essentially what we do is we think of the whole portfolio of collaborations that we offer as falling into four main areas. The first one is what we call devices, sensors, and mobility. And you've heard a bit about that, actually, if you attended Kristin Toll's Natural User Interfaces talk this morning, because the two areas overlap. It's the idea that most people have a cell phone these days, and those cell phones can have tremendous capabilities. They have high-quality screens. Naturally enough, they're always connected to a network because they're a cell phone. At least they should always be connected to a network. They have good computing processing power, lots of memory, considerable graphics capabilities. They're far more than is just required to talk to somebody as you would use a telephone. Now, if you imagine those capabilities combined with increasing bandwidth and high availability of networks, and you apply the combination of those technologies into other areas, you realize that this can become a tool for science or a tool for healthcare. And so there are plenty of projects going on which, just to pick one example, David Czar at Washington University in St. Louis in Missouri, he built a device which is an ultrasound probe. And the ultrasound probe actually plugs into a cell phone. So now what you have is you have an ultrasound device where you can scan someone with the probe, say a pregnant woman, to look at the development of the baby. And you can see the pictures on the cell phone directly. So now, instead of requiring a complex, portable system that might require an expert to use it, you can actually provide this to healthcare workers out in the field in areas where healthcare was not cost-effective to provide before. Because those images, when they're on a cell phone, they can be transmitted to a central location where an expert can analyze those images and transmit the results back. So the person wielding the ultrasound probe doesn't need to be an expert in what they're doing. And this can be cheap. So all of a sudden we have a way of scaling healthcare into regions that didn't have the ability to do it before. Now I just give one example here. There are many other examples, many other types of device you can extend a cell phone with, or many other capabilities that a cell phone has that you can use to deliver healthcare or do healthcare research around the world. So that's one area. Genomics in healthcare is a second area. The idea that even now, the human genome was fully sequenced only 10 years ago, and it took over $1 billion, and it took somewhere between 5 and 10 years, depending on where you think the project actually started. These days, however, it takes less than $5,000 and less than a week to sequence a human genome. So the technology has advanced hugely, and it hasn't stopped. There are increasingly large data sizes generated from machines with increasingly high capacity, and I believe that within 5 years, most people will be able to have their genome sequenced for less than $1,000, at which point, at least in the United States, it becomes quite cost-effective to sequence everybody who has any sort of healthcare coverage, which means that the amount of data will be huge and the requirement to understand exactly what that DNA is telling you greatly increases. So that's another area, and we're interested in the computational challenges around that. Medical imaging is a third area. We're just starting to develop into this area, but we have a range of Microsoft technologies which can take standard medical imaging like CT scans and automatically identify the organs that are represented on those scans. The state of the art today is that somebody has to actually look at these scans, and a human has to interpret what that scan is, what organs are represented on that scan. We have ways of automating that process, and we're starting to apply it now to things like screening people for colon cancer, or to determining how effectively an anti-cancer drug works by being able to scan a person's body, automatically detect all the tumors in that body, and look at how much they're shrinking in a very accurate manner to determine how effective a particular regime of drug therapy is for that patient. So we're starting to push the boundaries in that. We won't have any solid results for 1 or 2 years because research takes time, as you all know. The last area, modeling living systems, really is a combination of computer science and biological science. Biological systems are complicated networks of interactions between genes and proteins, RNA and DNA. And computers also are dynamic systems. And there are many different mathematical formalisms for modeling the performance of programs in computers, and of course Microsoft is very familiar with these. You can take those same formalisms, such as lambda calculus, and apply them to modeling a dynamic biological system, with the eventual aim of building predictive models. So you could do things, for example, like create an in silico model of a cancer tumor, treat it in silico with drugs, and determine which drugs are the most effective treatments for that cancer without doing any studies on a patient. And of course, doing it in a few minutes or a few seconds, rather than taking months to years and having all the ethical issues of testing those same drugs on patients. I should say we are many years away from that eventual goal. But we are starting to build dynamic models of biological systems, then looking at the issues of composing those models to build entire organs or even entire organisms inside a computer. That work's typically done in Microsoft Research Cambridge, but also in collaboration with COSBI, which is the Microsoft Research University of Trento Center for Synthetic and Systems Biology. So those are the different areas that we work in, and in each one of those areas we create these software projects that I described, and we create these collaborations that I described. Let me give you an example of how one of the collaborations works. Now, actually, although I've put up a slide here talking about the electronic contact lens, I'll only touch on it briefly because it was mentioned by Kristin Toll earlier today in the natural user interfaces, natural user interaction session. Essentially, what we did with this is Desni Tan is a researcher in Microsoft. He's very interested in human-computer interaction. You know, how you improve a computer so that you can interact with it in a simpler way with less constraints on the way you can interact. So you can be more expressive in working with a computer. So a computer might not look like the laptop I have in front of me. A computer could be built into this room. A computer could be anything. Now, there's another group in the University of Washington, which is also based in Seattle, so it's just down the road for us. And they're very good at microfabrication. They're very good at taking microelectronics and embedding them into unusual devices. What they wanted to do is they wanted to build a contact lens, okay? And in fact, they did, and you can see it over there. And if you have very good eyesight, you can actually see that there are microelectronics built into that contact lens. It's a soft contact lens. You can wear it like any other lens. However, the surface of the eye is covered with a liquid, and that liquid contains about 10% of the sugar level that is typical in your blood. So if you can watch how the blood sugar varies in the fluid on the eye, you can also know how blood sugar varies inside the body. And of course, if you're diabetic, that's a very important thing to know. Now, it'd be lovely if you could actually trace how your blood sugar varied through the day if you were diabetic without having to prick your fingers and take blood. And this could be done with this contact lens. Simply what you do with the contact lens is you build a little glucose sensor into the contact lens. You build some control circuits. You include a way of getting power to the contact lens, which it can be powered by radio waves. And you also put a little LED, a little light on the contact lens. And perhaps that light turns green if your blood sugar is okay, and it turns red if your blood sugar goes out of the normal range. And of course, at first, you'll notice this because it'll be a light flashing in your eye. But, you know, over time, it's pretty easy. You end up integrating these things into your natural senses. So, I mean, I wear glasses. I don't think about my glasses at all anymore. And the people in the audience who are wearing glasses today, they don't think about their glasses until I talk about it. Same with a contact lens. So after a while, what happens is that you don't see the red light, not consciously. You just go and eat some chocolate because you're kind of feeling like your blood sugar is low. But it's actually a sense that's being fed to you from your eyeball. So from Microsoft's perspective, this is valuable because now we have a computer that sits on your eyeball, a very unusual situation for human-computer interaction research. So Desni Tan, our human-computer interaction person, benefits from this collaboration. Now, University of Washington benefits from this collaboration because they get to build one of these contact lenses and test the proof of principle of their research. And so together, we're advancing the state of the art further than either of us could on their own, and we both gain new opportunities through this collaboration. So I said I wouldn't talk about this, and I have talked about it too much. So I'll just move on to another one. But we have plenty of other examples of this. One I'm involved in right now is a way of looking at drug clinical trials. Clinical trials are very expensive, they take a long time to run, and they require a lot of dedicated staff to produce all of the forms and the databases and the questionnaires and protect the privacy of all of the patients, all of that. And that's a big computing load on every clinical trial. We have a system that we've developed in conjunction with the University of Oxford that reduces that from being a whole staff of programmers for 6 months to generate that information. Now we can do it with one person in 2 days. So when we can do that, we can reduce the cost of a clinical trial. And now it's actually cost effective to test more drugs in the field more quickly than before. And because there's a whole semantic layer underneath that, if you saw Evelyn Villegas talk this morning, you'll know that Microsoft is heavily involved in semantic computing as well. Because there are semantics underneath, any clinical trial generated by this method, the data set is interoperable with any other clinical trial generated by this data set. So now, instead of each clinical trial being a single custom example of collecting data, they all feed into the same underlying body of data which continuously grows over time and provides more value to the medical community. So that's the second example. I said I wouldn't talk much about that, and I did. I'm sorry. Now I'm going to talk about MBF, and the rest of the talk is all about the Microsoft Biology Foundation, so we're now on topic. MBF is an example of one of those software developments I was talking about. So we work in producing these collaborations. That's one side of what we do. The other side is the software development side. And MBF is very simple. The principle behind it is very simple. It's a bioinformatics toolkit. It's a set of common functions that people can use to manipulate DNA, RNA, and protein. This is the basic building blocks of the rest of biology, basically. And the reason why we built a library of these functions is because we're just starting to move into the biological field, and so we chose the most basic building blocks of biology as the place to start. So that's what we did. Essentially, what MBF does is it provides you with only 3 or 4 different types of function. You can read files in biological formats. You can write files in biological formats. It has a bunch of biological algorithms built into it for the manipulation, comparison, and other types of changes to DNA, RNA, and protein sequences. And it also connects to web services. Microsoft clearly synthetic biology, so if you're studying pathways or metabolic flux analysis or biodiversity, all of these things are relevant to you. For example, for biodiversity, as I think probably the audience knows better than I do, very often DNA sequencing is the only way to determine the differences between closely related species of insect, for example, because the phenotypic variation is so limited. Or you might want to look at metagenomics and actually study the microbiological component of the ecosystems that you're looking at, in which case you would pick a different set of techniques, but again, it boils down to DNA sequencing. We do provide here an alternative to other biology frameworks. We know that there are other types of biology framework out there, BioPerl, BioJava, BioPython, and others. And they've been around longer than us. And they have a considerably larger selection of functions that they perform. However, what we found in bioinformatics is there are many, many different file formats, for example, because scientists like to define new file formats all the time, it seems. But only a few of them are used regularly. And so we, to a certain extent, starting late, or coming late into bioinformatics has provided us with an advantage, because many of the different file formats that have been generated, tried, and discarded in the past, we simply don't use. There are very few file formats which everybody uses, and we make sure MBF handles those formats and handles them very well. The other thing that we do well is we handle large data sets. When we were going to genomics institutes and finding out how they work to collect information to build MBF, what we found is that a lot of the time, the tools that worked very well 5 or 6 years ago in this field are no longer capable of handling the very large amounts of data that are now available. Somebody was giving me an example of how they ran a tool that they've been using for many years, and now it takes 3 or 4 days simply to load the data into that computational program that it used to take them 10 minutes when they first started out. So things aren't scaling very well here. And so we spent a lot of time thinking about this problem and figuring out how we handle very large data sets. A couple of examples of the way that we do it inside MBF. One of them is that if your DNA sequence, if you're interested in only variations in a DNA sequence and not the whole sequence, then we have a sparse sequence model, which enables us to store only those variations and not the sequence. Now, it's completely transparent to the user. You don't realize that the computer is only manipulating a small set of data rather than the large data set you fed it. But nonetheless, it's one way of improving the efficiency of the whole system. The second way is we know that a lot of biological data exists in extremely large files. I just downloaded one on my laptop today, and it's 960 megabytes, and that's compressed. and that's one of 5 or 6 of those files that I have to download. It's actually the smallest one of the set. So biological data sets are growing all the time. They're already very large. What we do in MBF is we actually have a virtualization layer built underneath all of our algorithms, so that instead of reading the whole file in one go, we actually take a chunk of that file, read it in, and then page other chunks in and out of memory as you walk through the file, so that you never actually load all the data. And that way, again, it can be more efficient. And there are other types of efficiency we've been building into the algorithms as we go, and Mike will be showing you some examples of how those efficiencies are actually improving the performance of MBF later on in this talk. The design goals of MBF. We wanted it to be extensible. Microsoft might have built MBF initially, but the purpose here is to build a community around MBF, to get other people to make contributions. We can't possibly build MBF to satisfy all of your scientific needs. We'd be doing it for decades. We'd never be finished. One thing about biology is, even if you have a room full of biologists who are working in the same biological area, they'll all be working on different aspects of the problem. They'll all require different programs. If you look at the number of scientific computing programs available out there for biology, there's a huge number. Each one of those programs only has a very small number of users, because it's only appropriate for a very small area of biology. Microsoft can't possibly build the thousands and thousands of tools that you all need. So what we've done instead is we've built a core of those common components, and that's MBF. But we look to the community to extend it to meet their own individual needs. And we've made it easy to extend, and that's the extensibility piece, and I can talk more about that later. Language neutrality. Not everybody programs in C#. It's a shame. I think personally everybody should program in C#. But if you wish to use other programming languages, the advantage of building MBF on top of the .NET platform is that .NET supports over 70 different languages. So if you're familiar with Pascal, or you like to use Visual Basic, or if you want to use a functional programming language like F#, all of these are supported. You can write in any of those things, and you can use MBF natively as if it was written in the language that you're using. And you can contribute to MBF in any of those languages and extend the functionality. So you're not tied down to using one particular language. The other thing is we've tried as best we could to ensure that the way in which MBF is written is also a lesson to how to write good code, because biologists aren't necessarily programmers. So when I was learning, when I was a biologist, I didn't pick up a book and read about a programming language. It's boring. I didn't want to do that. I wanted to do my job. So what I did is I downloaded code from the net, and I looked at how some other people had done similar things, and then I hacked that code until it did what I wanted it to do. And I expect most of the people in this room, especially if you're familiar with using Perl or Python or Java, you're probably familiar with doing that kind of thing. It's basically how bioinformaticians work. So what we did is we made sure that our code examples are extremely well documented. There's documentation in the code, so you don't have to spend your time hacking around in great detail to try and make something work, because you can read how it works in the code. It's an example of the best practices of how you would use C# in your application, so it's also a lesson in itself. It's an education just to read the code of MBF, and that's by design. Now, not only that, but we've implemented basic algorithms. Algorithms you can read in the literature, Smith, Waterman, Needleman, Wunsch, things like that. And every time we've implemented one of those algorithms in MBF, we've included the literature reference in the code as well. And so we're not doing anything in secret. If you want to know how we're doing pairwise comparison, you can go read the reference, and that's exactly what we implemented. So you'll learn from that as well. We've also developed training course materials available under the Creative Commons license, so that anybody can take those training materials. They include worked examples of the workshops, so that you can actually write code on your machine as you follow along. They're all available online because they're Creative Commons. You can download them, you can edit them, you can manipulate them in any way you like, and they actually form a good basis for an entire bioinformatics curriculum. And of course you have MBF, and MBF is available under the Apache 2.0 open source license. So again, you can offer that to students as well. So I'm just really making a point here that I've made before. MBF is not an application in itself. If you download MBF, you can't run MBF. It's not a program that you can run. MBF is a basic layer of functions, and you have to build tools on top of it. Now, we offer some of those tools. We actually offer some demo tools that allow you to assemble DNA sequence, to do comparative multiple sequence alignment, for example, to use some visualization so that you can see interesting associations between the sequence of interest and the blast hits that are returned from NCBI, for example. All of these things. They're not intended to be fully functional applications in their own right, although no doubt some biologists will find them useful. But they've been built in a way, many of them are open source. You can download the source and you can extend them to meet your exact needs. So, I'll skip that. This is something I often get asked, so I'm just going to cover it here. We're Microsoft. We'd like you to see the benefit in using Microsoft tools, but I'm not here to sell you a copy of SQL Server. We're in Microsoft Research. We're very fortunate in that the people that we work with, and the reason why we work with those people, it's all about the scientific research, not about selling copies of our products. But we want you to see the value in using Microsoft technologies for your science. But the science comes first. And if you need to use a different platform, that's possible. So what we do here is MBF uses .NET as its platform, but there's a public domain version of .NET called Mono. And Mono is implemented in such a way that it runs on Linux and it runs on OS X, so you can use it on a Mac. So what you can do is you can actually take MBF and you can run it on a Mac or on a Linux machine if you want to. Now, I don't guarantee it'll run perfectly on a Mac or a Linux machine. We don't test it extensively in those environments. And probably you'll find that it's faster and more flexible if you're running it on a Windows machine. But nonetheless, this is an option that's open to you. Just in the same way as we parse a huge variety of file formats and we build APIs so you can build parsers for whatever files you want. Just in the same ways we connect to web services so that you can access data sets which aren't accessible to us, perhaps even aren't on the Microsoft platform somewhere there. Interoperability is not just a good idea. We need to be interoperable to survive in the scientific community, and MBF is interoperable. So don't think that you're just forced to use Microsoft all the way. Although if anybody would like to buy a copy of SQL Server, please come and see me at the end. Okay. So, I've talked about MBF being open, I've talked about it being open source, and the last real step here that I should talk about is that it needs to be a part of the community. We need your help, because we can't build everything we need. And so what we're doing is we've made it available for commercial and academic use, free of charge, under the Apache 2.0 license, so anybody can obtain it. The installers are available, as you'll see from that URL there, and the source code is available from CodePlex. Now, CodePlex, if you're familiar with SourceForge, you're familiar with CodePlex. It's a repository of open source code. So you can go there, you can download the source, and you can do anything you like with it. But more than that, we're building a community around this. So we're offering training courses free of charge. We're putting those training courses as videos on the web. We've got a set of forums. If you go to mbf.codeplex.com, you can actually submit questions and the development team will answer them. If you find bugs, you can submit bugs and the development team will fix them if they have the time. And of course, you can become contributors. And if MBF doesn't do what you need it to do, please write the extension that you need. Send it back to us and we will integrate it into the code and make it available in the next MBF release. And it will, again, be under the Apache 2.0 license, so you're not giving something to Microsoft particularly. You're actually contributing it to a growing MBF community. The other thing I should say is the MBF project is not guided by Microsoft alone. Since the beginning of the project, we've been guided by a technical advisory board in which Microsoft is a member, but we also have other adopters of MBF on that board. We've got the University of Texas at Austin, we've got Queensland University of Technology, we've got Cornell, we've got Illumina Corporation, manufacturers of one of the most well-known DNA sequencing instruments, and we've got Johnson & Johnson Pharmaceutical Research and Development. So we have an excellent mixture of commercial and academic adopters actually steering this project into the future. We don't say what goes into MBF next, they do. The community says what goes into MBF next. And the last thing here might come as a surprise to you. MBF will not be owned by Microsoft. Microsoft has built it, most of it, to date. We've been responsible for most of that. But as soon as version 2 is released in July, one of the announcements which will occur when we release is that Microsoft has given up ownership of MBF, and we've transferred it to the Outer Curve Foundation. Now, the Outer Curve Foundation logo is here, and actually Outer Curve is similar to the Apache Foundation, if you're familiar with the open source community. It's basically, it is an entity that exists to own software projects. And because they own it, we no longer own it. We want to be contributors to the community. Microsoft wants to help build MBF. We need to see it as a success in the future. Our own internal Microsoft researchers are using MBF in their own research, and they will be making contributions into MBF in the future. But what we really want you to do, and if you don't take anything else away from this talk, please take this one thing. We would like you to become contributors to it and to extend it yourselves to make it useful to you and to contribute also to the community and make this a success. So thank you very much. I'd like to introduce you to Michael Ziskowski, who'll now give you the second part of our talk.
Cool. Thanks, Simon. You guys can hear me okay?
Oh, yeah.
Awesome. Thank you, sir. Okay. So I'm the program manager on this project, and quick background, we're almost through the second year of effort on the Microsoft Biology Foundation, MBF. I'll talk briefly about what happened in the first iteration of the project, but most of the rest of the talk, and I promise to get you guys out of here in time for lunch, will be about what's happened the later part of this year and what the advancements are in the software library that you can expect in July. Okay, recapping, and I'm going to go pretty quickly through some of these slides to make sure that we get out on time. But a lot of this is what Simon already mentioned. We're going to support the genomics domain from the sequencing machine up into a fully annotated genome. And that's the scope of the effort to date. Again, any library contributions and any further domain interest, whether proteomics or other areas of functional genomics, it's all possible. But this is the area that we're focusing on to give a really solid foundation to the library and allow it room to grow. We found that the customers, the researchers in this space, really vary widely in their expertise from the biology through the computer science. You know, bioinformatics is a relatively young discipline, and so we've tried to be as complete as possible in all of the documentation, in the coding styles, in the code comments, and make it as easy as we could for the novice computer programmer to come up to speed quickly. So if you're an advanced computer science programmer and you've been around a while, and you'll find it a piece of cake. But we really wanted to go for the least common denominator of the types of people who would likely be trying to use the software. And we also, you know, the tools side of the equation are also pretty important because it's really hard to express the value of a software library without showing what it can do. So we have put some effort into some of the tools, and in doing so, we've tried to pick out some of the more interesting or common scenarios that the biologists and the bioinformaticians are doing in this space, and give a good view of what the library is capable of doing in this regard. And so this is also part of our audience, is the actual biologists who are using tools that are built on top of this platform. So whether you're more on the computer science side or more on the biology side, I hope that I can address all of your needs. Okay, so high level, Microsoft Biology Foundation consists of an object model, which is essentially the representation of genomic sequence data. On top of that, you've got more of the functional components of the library, including, you know, file parsers and formatters. This is how you get your data in and out of the object model. The algorithms that allow you to do alignments, assembly. You can do transcriptions, and then the web service interfaces. On top of that stack, then you'll have the tool sets. And we're providing a couple pretty compelling examples, I think, of how the library can be integrated with Excel. And we have a self-contained application called a sequence assembler. It's written in .NET and WPF, and it shows some of the visualization capabilities that you can get by integrating different Microsoft technologies together. And then some command line tools, which are really just simple samples, functional programming tools for leveraging the capabilities inside the library. Okay, getting a little bit deeper now. These are the namespaces we're currently using inside the library. The base name we start with is bio, and this is again kind of matches to the object model I showed on the previous slide. And we have the concept of sequences, DNA, RNA, proteins, the different alphabets, right? Ambiguity characters are supported. We also have single nucleotide polymorphism representation inside the library. Base level, highly efficient data structures, which is a really important thing when you're dealing with very large sequence data. Memory consumption is a critical factor in maintaining performance and the ability to operate on large sequences. The I/O portion, these are a set of file format extensions that are currently supported in the library. If you're familiar with these, they'll make sense. If not, they're basically the most commonly used formats for sequence representation. And there's a whole bunch of different ones. So in addition to picking what we heard from the community as the most widely used, we also made it very easy to extend the format and add any other types of file format supports that you might need. The one in italics at the bottom has actually been added for version 2. We're focusing more on annotation support for the next release of MBF, and this is part of that effort. The algorithms, we started with alignment algorithms. These were kind of, we found the base need of most researchers in this field. Smith, Waterman, Needleman, Wunsch. We have implemented MUMMR and NUCMR, which are pretty widely used alignment tools. And then MSA is a multiple sequence alignment novel approach based off of a few other algorithms in the industry, but done by a researcher that we had from Carnegie Mellon University. And he wrote a paper and published that. And so we have, in addition to doing the industry standard sort of implementation of algorithms, we're also applying some new techniques that we think could improve the capacity, quality, and performance of alignments. Assembly, de novo assembly is used to piece together DNA reads into a contiguous sequence without a known reference, and this was something that was implemented in the V1 effort for MBF. Since that time, we've made great strides in increasing the capacity capability of the library. When the first version came out, we really took a breadth approach. And our motto was, we're going to reinvent the wheel so you don't have to. Try to give a base amount of functionality that had some degree of parity with the BioPerl and BioJava libraries that are available. But we didn't go very deep, and we didn't scale very well. And so the biggest effort, the largest effort that we're taking in the current implementation of the library is to increase the capacity and the performance and the scalability of the library. So the de novo assembler has come a long way, and I'll show some numbers here in a few minutes. Comparative assembly is a new feature inside the library, and this is, the concept here is that, well, actually I'll address that in a second. And then web services, primarily the ones we've implemented are against BLAST, different BLAST services. But again, the framework supports easy extensibility to all sorts of different web service connectors. Okay, so assembly of large genomes. I think I mentioned this one. That's our top goal right now. We're also focusing more on the annotation side. So being able to read different formats of data that contain annotations, allow user annotations, and support in tools that also provide for some visualization capabilities, and I'll do a quick demo. We also want to make sure that the developer experience is very good. So debugging is easy, easy to create and extend new parsers, algorithms, well documented, and easy to build tools on top of as well. I guess the other thing I should say about the developer piece is one of the things that we found by using the .NET Framework 4 is how easy it is to parallelize algorithms that lend themselves to parallelization via the .NET parallel extensions. And we've made extensive use of that, and especially in our assembly and alignment algorithms. And we think that's also something that people are going to appreciate. And community is the big one. So this is really, Microsoft is in this just to get it going. We really want this to be a community-owned and led effort. It's really up to the community to decide how widely this is adopted and used and how it expands. And we're going to continue supporting the community, but we're very adamant that that's the direction this is going. Okay, so one of the techniques we use at Microsoft when we do software development is we identify scenarios that are core to what the software experience should provide at the end. This helps define and scope our effort. It also helps us know when we're successful, and it keeps us focused along the way. So our core scenario that we chose to help address the primary goal of supporting large genomes in this effort was taking some example polyploid genomes, agricultural genomes, and making sure that we can assemble those. And it's one of the reasons that we've implemented the comparative assembly techniques that I'll show you some details on in a minute. Lots of different ways that being able to assemble sequences of this nature can benefit society. We have an internal science team at Microsoft Research and led by a man named David Heckerman. Rick Rashid briefly mentioned him in his opening keynote and kind of skipped over a few things. But he's pretty well known in the field, in the software development field for using machine learning to create the spam filter in Microsoft Outlook. Well, he also happens to be a PhD MD and is now applying those machine learning techniques towards the analysis of HIV. And so he's an internal customer at Microsoft that we have very close access to, that's well connected with the rest of the bioinformatics community, but also provides us with a really valuable scenario with data access and the expertise to tell us if it's working or not. So we work closely with David and his team. We align our goals against the scenario. And our key deliverables are to make sure that we actually support the assembly of large genomes so that it can be leveraged in other ways. And then also the analysis tools that can be used by the scientists to evaluate how things came out and do annotations. And there's some pretty cool things that can be done using deep zoom and some other things that you might have seen earlier. Okay, so de novo assembly, I'm going to assume most people understand what that is. It's been around for a while. The comparative assembly process, I'm going to show in detail on the next slide. The point here is that reads come off a sequencing machine, and then the reads are they have some filtering done on them. Make sure there's good information. You throw out the bad reads, and then you can bring them into the library and start working with the data. And you have to reassemble all these reads back into what is a contiguous sequence of DNA, and then you can start figuring out, well, what is this person like? And then that part happens at the end. So this is a busy slide, and I'll try to walk through it quickly. There's a data preparation step at the beginning. That's what I mentioned a second ago. And that's something that's outside of what MBF can manage right now. That's something that happens more close to the sequencer. Different sequencers have different filtering requirements. They have different error rates. They use different chemical processing or image processing to generate the reads, and so it's very that part of the process is very specific to what the instrumentation is that you're using to collect DNA information. But after that, it gets pretty, the workflow is pretty common across most bioinformatics problems. We have, and what you see here in the boxes are the command line utilities that are provided with the NBF platform. And it's a way to exercise the library to support this workflow. The Padina util, Padina is the name we gave the de novo assembler inside NBF. It's the parallelized de novo assembler. So it uses the .NET Parallel extensions to make efficient use of all the cores inside your machine. And I won't go into too much detail here, but for the comparative side, it's a pretty interesting technique. And essentially, the idea is that you're taking the reads, but you're using a reference. It's not de novo, which means against no reference, but you're actually using a reference genome that's a similar species. So if you're trying to sequence a genome that hasn't been sequenced before, you don't have an exact reference. But you might have something that is closely related, that's perhaps mutated over time via evolution or maybe short spans of evolution, but you're able to use that as a reference to help guide the rest of the assembly process. And the tools used, there's a whole sequence of steps that happen, and we provide one tool called Compare Util that actually will do the whole thing for you. But we've also provided atomic unit operations so that researchers can perform one step, evaluate results, become comfortable with what it's doing, maybe make changes to the code or maybe make changes to the parameters that go into that algorithm. But we try to make it as easy as possible for people to use in whatever steps and sequence of events that they need. So a read alignment happens. There's some optional steps where if you have paired end read information, you can actually optimize some of the matching information inside the read alignments. And then there's a consensus generation step that happens, and there's a separate utility for that. And then the scaffold generation, which actually puts together the various contigs of sequences and aligns them against, what is assumed to be the final DNA strand. But the only step that uses the reference sequence is the first one. And then the rest of it is all done against itself. So it's a really interesting process and relatively novel. And we've released a beta. Simon mentioned we're going to release the final library in July. And I have some kind of hot off the press numbers that I'll show quickly, but they will change by the time we go out the door. One of the things that we've been doing is we've been using the E. coli MG1655 DNA sequence as a really good benchmark. There's a lot published on this genome, and I realize it's not large, but we have a lot of information that we can compare how the NBF library behaves against other libraries that are out there. And I think this is an area that I get a lot of questions about. So we've got a native implementation of Mummer on Windows on the same machine, and we're running we call this our super desktop. It's a 24-core Windows 7 machine, 64-bit with 196 megabytes. Sorry, that should be gigabytes. That's an error. 196 gigabytes of RAM. So it's a big machine, but it wasn't really that expensive, and it wasn't really that hard for us to find. And when we talked to the labs that do DNA sequencing, this was actually a mid-range machine that they typically use. So we feel pretty good about the configuration that we're using to test these things. And these on the bottom scale here is the coverage of reads against the genome. If you're familiar with that, or if you're not familiar with that, that's the oversampling basically that you have on a specific sequence of reads to help you identify either errors in the reads or where you have gaps in alignment. And the higher level of coverage basically means you have a better chance at assembling a genome together. The data sizes also go up dramatically. So basically what we're seeing here is a really good improvement in terms of the MUM-util tool in NBF written in the .NET language against a native implementation of Mummer in Windows. And especially as the data sizes go up, the MBF library scales very well against a native implementation, mainly because this is a single-threaded implementation. And the use of parallel extensions in MBF library scale well across the 24 cores on the machine. So total transparency, but it shows the value of what the library can provide. We're also working closely with, as Simon said, multiple people in the community, but Cornell University has also been doing a lot of benchmark analysis on their own. And we were happy to see that from our V1 release of the library to our V2 release, on the same E. coli strand that I just sent you, they saw a pretty significant improvement in time to process that genome as well. And this data was on the beta release of MBF, so you guys can download it yourself and try this yourself and validate. Oh, that's a mess. Okay. I will probably just skip this slide. This is the, and this is apples to oranges anyway. I tried to be local in my use of analogies, but this is a benchmark paper that we are going to be using, and we will be publishing the MBF results against the benchmark analysis here done with different de novo assemblers. Right now, we're looking really good. You can't really see the numbers, but I think the main point is that we're trying to be very upfront with, well, how does the capabilities inside this library stack up against the other things and the other tools that you might already be using. Okay, we've got a couple other, I mentioned the HIV work that David Heckerman and his team is doing. The nice thing about this collaboration is that they're a little bit ahead of us in terms of the advanced research techniques they're doing. And so when they find and identify some useful nuggets, then we'll pull those into the library. And one of those is this biology matrix class. And they've also got a distributed computing library that they've been working with for a while that we're also going to provide in the library now for more general use. So it'll be easy to not only parallelize your applications across cores, but you'll also have a really easy framework to use on HPC clusters and possibly going up to Azure as well. I'm going to skip over this. This talks about the biomatrix specifics. The community challenge, we've talked about a lot already. The key thing here is that we're going to facilitate responses to community requests. We're going to invest in people's education about the library. We just held a 2-day workshop over at the Santa Clara Hotel this week on Monday and Tuesday. Very well attended and had a lot of good questions and answers there. And it's an open source project. Through and through, it's free. We're not trying to sell anything. We're stepping really out of the leader's seat and we're generating the leadership from inside the community of people who find this valuable. And we want to continue fostering that. And I hope, and I'll reiterate what Simon said, anybody in the room who's interested in this at all and finds any value in this at all, please let us know, let the community know, get involved in any way that you think could be valuable. And the name is going to change, but for now it's still mbf.coplex.com, and that's the best place to start. Few closing remarks. We're also working closely with the Colombian government and Calencias on this bioinformatics computing center. We're really excited about what they're doing in this space, and we think that the MBF library, and as well as some other Microsoft technologies, can really help them get off the ground. And we've made a commitment to help them do this. And I expect that they will also become leaders and future drivers of where this platform goes, because the more people are leveraging it, the more vested interest they're going to have in where it leads. And I think that's great. Surging to the cloud is another opportunity. We've worked with another team at Microsoft on this thing called Azure Blast, and it's essentially hosting the Blast databases and the Blast query search engine in Azure. And and able to scale across multiple nodes in order to do quick, efficient BLAST queries. And it's something that can be hosted locally, or it could be hosted locally, but in the cloud, and then also offered as a service. So the future of bioinformatics and where I think this center going in stages is really, supporting research, developing tools that are needed to solve hard research problems, leveraging cloud-based resources, and then making services available to others. And that progression of things is a real natural one, and I think can benefit Latin America in particular. There's a few tools. I was going to show a demo, and I will if anybody wants to stick around. But I think the last thing I want to end on is letting you know that there's also a contest that's been announced. And it's really easy. All you have to do is use MBF and build a tool, and that does whatever it is that you would need to do in your work. And it doesn't have to be fancy, and it doesn't have to be big. It can be as simple and efficient as you like. But we're hosting this now. It's open until July 15th, so there's still plenty of time. And the winner gets an Xbox and a Kinect, and then you can either play games or you can start doing some crazy augmented reality stuff like the guy from this morning. But I guess with that, if you have any other questions, I'll come back to this in a minute. But I want to give people a chance to go to lunch. And if you have any questions, I'm happy to answer those as well. Thanks. Thanks. Sure.
Thank you. Just two brief questions. The first one is that in the first part of the talk, Simon talked about one of the focuses of Microsoft Research or Microsoft Connection is the work on the building of biological computational models of biological phenomena. But it is clear that MBF is focused on DNA, RNA, and protein sequences and analysis and stuff. So it is not integrated. I mean, are there any plans of integrating, or better, how is Microsoft working on the building of applications, or is there any framework in that part of modeling biological phenomena? And the second one that is out of curiosity, why not supporting from the very beginning of MBF the MBL format?
Well, do you want me to get that?
Sure.
Okay. To answer your second part first, if you'd like file format supported by MBF, you'll be very pleased to know that you too could become a member of our community and provide that support. Unfortunately, that's going to be the answer to a lot of questions in terms of the functionality that MBF has. We provided a starter set of functionality, which relates to the scenarios that Mike was showing, the ones that we've been particularly targeted. But we don't pretend that this is a comprehensive coverage of everything you could do. And so what we've done is we've provided the APIs and the documentation that enables people to extend the library themselves. If you do extend it to cover that particular file format, please contribute it back, and then we can incorporate it and make it available for everyone. But that's pretty much how it has to go, because we can't satisfy everybody. Now, in terms of the modeling work, the modeling work actually is mostly occurring in Microsoft Research Cambridge, and they started many years before MBF. And so there is no direct link between the two at the moment. I talked about that more as an example of one of the many projects that we're doing, rather than related specifically to MBF. Now, the modeling platform that I'm most familiar with that they offer is called SPIM, S-P-I-M, the Stochastic Pi Machine. It's downloadable from Microsoft Research Cambridge. And the second one that's based on a similar system is called BLENX, B-L-E-N-X. And that is available from the University of Trento Center that I mentioned. You can find links through the Microsoft Research Cambridge site. These are tools that exist rather than MBF, which is a library of functions, so it's not necessarily possible to get into it and program it yourself, but it will run there's a simple modeling language you can follow, and then you can use this to run your models. Okay, does that answer your question? Okay.
Cool.
Just to sort of dig into that a little more, I'm just sort of interested to know when in version 2, when this I understand the value of this is going to be the community contributions, but I'm interested to know what you're going to contribute going forward. Are you going to move into the proteomic space, or are there plans for that? That's sort of my big question. My small question is, is the benchmark data you use actually part of the library that's downloadable?
I'll take the benchmark question. I'll let you talk about me next. No, but it's a great idea. I think we should. I mean, it's publicly available on the internet in various places, and we have the links and the documentation. But I think it makes sense to include.
I mean, that association is, I think the field is littered with cases where people claim it's not, one thing or another, but you can never actually test it because you can never get the benchmark data.
Perfect. Yeah, that's a great idea. I'll take that back.
I'm guilty of that myself, I should say.
Yeah.
No, that's a great idea.
Yeah, I mean, just on the benchmarking stuff, NBF actually comes with a set of unit tests and the data required to support those. So in theory, not quite true in practice, but almost all of the code is covered with an extensive range of tests so that you can show how accurate it is, and the data necessary for those tests does ship with NBF. Now, the benchmarking stuff you saw here relates either to an E. coli strain that's already the subject of a publication. So I don't know whether we would actually wish to provide it separately, but we should certainly provide the accession number for that. We'll make sure that happens.
Yeah, the only downside is the size. I think it's like a 7 gigabyte download, and we're trying to balance that.
Right.
But it's at least and actually the benchmark metric tool we're using was provided. There was an assemblathon effort down in California earlier this year, and that's what we're actually using to generate the benchmark numbers as well.
Right, and we were thinking of using the assemblathon data sets as well, which is what I was going to say. They tend to come in at about 5 gig each, so I'm not sure that we would want to ship those exactly, but because they are standards or
maybe
intended
provide
to be standards
separately
in the community, we'll certainly explore that.
Yeah.
On the proteomics side, what do we plan to do next? Well, that's a very good question. I'm glad you asked. I don't think proteomics is a part of what we plan to do next, as a matter of fact. But although Microsoft is stepping away from leading and owning the software and leading the community, it doesn't mean we won't stay as active contributors. We will. And there's several areas that we're looking at. One is the David Heckerman's eScience team, part of Microsoft Research, as I said before, is using the library as part of their research. There are several different tools and technologies that they've been producing that we're going to absorb into the library over time. Typically, the way it works is David's team produces an application and then produces some publications based on it. I know very recently they published some work on using fast Fourier transform in genome-wide association studies. That work and other similar work will be absorbed into the library over the coming year so that we'll be able to say that future versions of their tools will be entirely based on NBF. So that's interesting in many ways, but the principal one that's of interest to us is that you could go out and use BioJava today instead of NBF because we have the same functions. But in the future, NBF is going to actually be able to do things based on unique Microsoft research that no other library has, and that's where we want to get to with that. So that's one strand. The second one is, although the details are far from certain, we are going to be supporting the Columbia Bioinformatics Center, and we're not only going to be providing moral support, we're actually going to be providing practical programming development effort. And one of the things that we did on Monday and Tuesday this week when we met with the people from that center is we asked them to provide us with their needs so that we actually understand what things we need to implement next. and we'll develop those post version 2 and integrate those into MBF as well. So I don't exactly know what they'll be. If they turn around and say proteomics is what I need, then I guess that's what we'll be doing next. Okay. Okay.
Okay, great. I think that's it. Thank you all for coming. If anybody, actually, let me say one last thing. On tomorrow, Friday, from 10 to 1, I think, is our demo fest, and I'll go ahead and show both GenoZoom, which is a genome browser we're going to be providing, as well as some other tools that come along with the library. So please come then, and I'll be happy to talk more after the event as well.
Okay, thank you.